Hello everyone. So again a new day, a new beginning, a new thought. What we are today is due to the thoughts which make us. So it came in my mind, dear students, welcome to the new students who have joined or who are seeing my channel, the Science Funda, and the old students, because you can see me on the screen, I can't see the subscribers. I would love that I can get maximum subscribers because knowledge increases by sharing. Here I am just giving you some tips for last minute revision, and you are the receiver. Like we are forming a symbiotic relationship, if you agree. Give and take. It's a reciprocal relationship, right? So with all your enthusiasm and with all your good wishes, I start the lesson. What you can see in my hand. What do you see in my hand? What do you see? What do you see? What do you see? Your answer will be, we see what? The different shapes and the size of the very important organ of the green plant, the leaves. The leaves. Okay. Now, if it is the leaves, then what do you think we should go for? Because leaf is considered the kitchen of the plant, right? So like in your kitchen, what is made? Food is made. All delicious food is made. And we are heterotrophs. We cannot make our own food. We have to depend on some other organism for the food. And that organism on the earth, which can make its own food and is totally independent of any other organism, is nothing but the green plants. Okay? So, and that is all about today. The main points which you will do in this lesson will be what is photosynthesis, where it takes place in the plant, what is a tissue having so much characteristic feature which makes it the best site of photosynthesis, what are the factors which control photosynthesis, the internal factors, the external factors, then what are the two stages of photosynthesis, and what are the products formed and where are they used. And in the end of your lesson, I'm mainly focusing on ICSE syllabus. The experiments are given where you have to read, write and draw all the experiments. Okay, so let's go and start the lesson. Photosynthesis. First thing here you all should know is the definition of photosynthesis. So the word photosynthesis is nothing, photosynthesis. It is nothing but an event, a biochemical event or a physiological event, because this is a lesson of physiology, where the entire process takes place in a sequential manner. The solar energy is trapped by the green plants and which is converted into the chemical energy, and the food which is made in the form of glucose is stored as starch, and the plant releases very important byproduct, oxygen, which goes into the nature, and the important carbon cycle is formed. So in your book the definition is very nicely given. Synthesis of food from carbon dioxide and water in the presence of a very, very important pigment. Like we have a very important pigment in our body which makes our blood red. That pigment is hemoglobin. Here what is the pigment? The pigment is chlorophyll. The very important pigment chlorophyll, and for this only you see the green color of the plants in presence of the pigment chlorophyll, water and CO2. Now this was what is photosynthesis. Okay, now this photosynthesis, where does it take place? Where does it take place? It takes place in a very, very important organ of the plant. If you remember the plant cell and the animal cell, the plant cell contains a very important organ which is not found in the animal cell, and that is, or organelle I will say, not organ, organelle. And what is that organelle? The plastid. Plastid. And this plastid can be of two types, the colored plastid and the colorless plastid. A basic question. Two types of plastid, colored plastid and colorless plastid. So the colored plastid are known by the word chloroplast. Chloroplast. And this chloroplast is a very, very important organelle of the plant cell. Now this chloroplast is having a very complicated structure. The structure is given in the ICSE book on page number 70, figure 6.1. This is just a rough diagram I have made to make you understand. It's a disc-like structure. It is having two layers. It is an autonomous organ. It has its own DNA. The DNA synthesis takes place here on its own, like mitochondria and chloroplast are the two cell organelles where the DNA synthesis takes place on its own. So it is a bilayered structure. First thing, if they ask you write a short note on chloroplast, this chloroplast is having some coin-like structures placed one above the other. These structures are known, in general, these sac-like structures are known by the word thylakoids. Thylakoids. And when these thylakoids are placed one above the other, they are known as grana. Grana. And it is in this grana that the first part of photosynthesis takes place, which is the photochemical event and also known as the light reaction of photosynthesis, which takes place in the grana. Now, what is this matrix called as? This proteinaceous matrix. This matrix is known by the word stroma. What it is called? Stroma. And what takes place in stroma? The dark reaction, where the synthesis of carbohydrates takes place. Right? So these two reactions, the light reaction and the dark reaction, are the two very important events of photosynthesis. That equation which you see in your book in just one line, 6CO2 plus 12H2O making glucose C6H12O6 plus water plus oxygen, is a very simple reaction which you see, but actually it is not that simple. It is divided into two stages which we will see. I was telling you about chloroplast. Hope you will draw this diagram, draw a neat diagram of chloroplast and give the features of chloroplast. Gave you that. Now after this, another important pigment which is found in the green plants exclusively, whether it is a gymnosperm, whether it is an angiosperm, whether it is algae, because all algae are autotrophs. Okay, remember all the green except fungi. If fungi, if you know the plant kingdom, how the plant kingdom is divided into. Okay, the gymnosperm, the angiosperm, and the lower group, the higher group. We are talking here about photosynthesis, and I'm talking about chlorophyll now. I'm now talking about the two main pigments which is required in photosynthesis. The main pigments which show their role in photosynthesis are chlorophyll, first you will write, and another one is carotenoid. Now this chlorophyll is of following types: chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, chlorophyll E. If you have studied in your previous class, or if you know the word chromatography, chromatography is the technique through which these colored pigments are separated on a chromatographic paper. We separate these pigments of chlorophyll, we take the leaf, we crush it, and we squeeze out that extract, that chlorophyll extract. Then we take a chromatographic paper, draw a line over it, and separate the pigments of chlorophyll. It's a very nice experiment if your teachers can show you on a chromatographic paper the pigments of chlorophyll. Okay, and this chlorophyll, it absorbs the wavelength of light, visible wavelength of light between 400 to 700, right? And maximum light absorbed is the red light, the blue light, and the violet light. The green light is not absorbed, it is reflected back, so the plant looks green. So the maximum photosynthesis takes place in these three lights, okay, the three wavelengths, red, blue and violet. So chlorophyll does this job. Now the carotenoids, they help the chlorophyll to trap the solar energy and also prevents it from photooxidation. The carotenoids are of two types, okay, xanthophyll and carotene, the colored pigments which you see, like you see the colored vegetables, some colored plants. What are they? Those pigments, what are they? They are nothing but the carotenoids in them. So the first raw material used for photosynthesis is the chlorophyll. Where it is found? It is found in chloroplast. What is the structure of chloroplast? Bilayered structure. It's a macromolecule. It is having stroma. It is having grana. In the grana, the light reaction. In the stroma, the dark reaction. Okay. Now, where is this chloroplast distributed in the plant? Where? The question comes. Now, open your book and turn to the page 72. A neat and clean diagram is given because biology without diagram is incomplete. So, you have to draw this neat and labeled diagram to study the distribution of chloroplast. I have drawn the rough diagram here. Okay. Now this is the part of a leaf. Now the upper epidermis. Then the two layers which are written here in your book also you can see. One is the palisade cells, What is the spongy cells? The spongy cells are irregularly arranged, and the palisade cells, they both are studded with the chloroplast. Chloroplast. They have the chlorophyll in them. And this together, palisade cell plus spongy cell, together is known as the mesophyll tissue of the plant. So the photosynthesis is mainly concentrated in the mesophyll tissue of the plant. First point. Second point, this chloroplast is also found in the stomata. If you know the structure of stomata of your 9th standard, and also if you have studied this stomata in where the root lesson was there, the lesson transpiration was there, where you see how stomata plays a role in transpiration, gaseous exchange and photosynthesis. And so the stomata are found in the guard cells, which are the kidney shaped cells here, and the chloroplasts are found here also. So the photosynthesis takes place in these guard cells, both in the daytime and in the night time. Okay, this is the previous knowledge which you should know. Now in the daytime, bright sunlight is there. So in bright sunlight, what happens? ATP synthesis is there. This is the energy currency of the cell. ATP, adenosine triphosphate. This is there. Its production takes place in the light reaction. So when this production of ATP takes place in the light reaction, the name of ATP is adenosine triphosphate. As I told you, photosynthesis is divided into two parts, light reaction and dark reaction, which I will tell you. But before that, the mechanism of the mechanism of open stomata and a closed stomata. Open stomata and a closed stomata. So, when does the stomata open? What is the mechanism which is governing it? In your book there are two mechanisms given, the potassium ion theory and the sugar theory. So, to understand that, even the cycle is very nicely given in your book, if you can see on page number 71, where they have shown you the opening and closing of stomata based on the potassium exchange theory. Now what happens here in the daytime? In the daytime, these guard cells, which are kidney shaped cells, they show high concentration of potassium. Why? Because ATP synthesis takes place. High concentration means it becomes highly hypertonic. When it becomes hypertonic, it tries to draw water from the neighboring cells. That is, endoosmosis takes place. When endoosmosis takes place, stoma opens, the stomatal pore opens, okay? And the guard cells become turgid. So the keywords are high concentration of potassium in the daytime, endoosmosis, hypertonic, turgidity, stomata opens, the guard cells open. The reverse takes place in the night time. What happens in the night time? The guard cells become flaccid. Here in the night time, ATP production is not there. Here what happens there? ATP is utilized for the potassium ion to efflux. Efflux means to go out. In the daytime, influx. The word influx is given in your book. And efflux is going out. So in the night time, what happens? ATP is utilized. For that, the potassium ion has to come out. For that, the concentration within the guard cell becomes hypotonic. For that, exoosmosis. For that, flaccid. So these are the sequential things which happen during the daytime and nighttime, the mechanism behind the opening of stomata. This was, if they ask you, explain the potassium ion theory. You can make a flow chart given of your book, and what I told you, LMR. On your right hand is day, left hand is night. So daytime what happened, nighttime what happened. Now the next theory which is given in your book is the sugar concentration theory. What is the sugar concentration theory? This is telling that according to the old sugar concentration theory, during daytime the guard cells begin photosynthesis. Obviously daytime photosynthesis. And what happens? The sugar concentration increases in the daytime. The osmotic pressure increases. So as the osmotic pressure increases, because sugar is a solute, it increases, the guard cells tends to draw water from the neighboring cells, and endoosmosis takes place. And thus the cells become turgid and they bulge outward, and the widening of the stomatal opening. Got it? Two things. And remember, if they ask you what are the features of the guard cells, it is very important to know the specific feature of the guard cells. They have thick inner wall and which is facing the stomata, and the thin outer wall opposite to it. Thick inner wall. That is, I have drawn this thick inner wall, and the outer walls are thin. So till now, what I told you what is photosynthesis. I told you where it takes place, mesophyll tissue. What is mesophyll tissue made up of? Palisade layer and spongy layer. Now, after that I told you what is chloroplast. Chloroplast is this structure made up of grana, stroma, thylakoids. What takes place in grana? Light reaction. What takes place in stroma? Dark reaction. And then I told you the mechanism of stomatal opening in the daytime and the nighttime. This was the background of photosynthesis. Got it? Now we will now come to the main part of photosynthesis. Now there is one part in your lesson, page number 73, where it is given how the leaf is adapted to photosynthesis. Now, first thing is, like if you see here, see this is the big leaf. It's a big leaf. Okay, this is a creeper. You see, this is a creeper. Creeper I brought. This is another plant I brought. This is see the shape of the leaf. See the shape of this leaf, see this shape of this leaf, just see, and see the shape of this leaf. What is the common feature between all of them? What is the common feature between all of them? They are green in color. Okay, they are green in color because they show photosynthesis. So you please come to your page number 73, where they have given you the factors which affect photosynthesis. Some are the internal factors and some are the external factors. In the internal factors come the surface area of the leaf. So what is it? Larger the surface area, maximum will be the light absorption. So this big leaf will show more photosynthesis as compared to this one, and this will show larger than as compared to this one. Got it? So this is just like a chain of events which takes place in the nature. And that is why the plants are of various types. The herbs, the shrubs, the creepers, the climbers, the trees, which you know that. How the plants are divided into. Okay, the gymnosperm and the angiosperm. So it is the surface area of the leaf which governs the rate of photosynthesis. Second is how the leaves are arranged in the plant. If they are at right angle, maximum absorption of sunlight takes place. The arrangement of the leaves on the stem, how they are arranged in the plant. Third is the cuticle and the upper epidermis, which is the transparent and a waterproof layer which allows the light to enter in. This sunlight to enter here. This epidermis layer is a waterproof layer which allows the light to enter in. And it is made up of cutin. C-U-T-I-N, cutin. What is epidermal cell made up of? Cutin. Okay, that chemical it's made up of. So next important requirement is it has numerous stomata. It should have numerous stomata. Around 500 to 5,000 stomatas are there in the leaf, so that maximum photosynthesis takes place. Then the leaf should have the thinness of leaf, because the thinner the leaf, it reduces the transport of water between the cells. Rapid transport takes place. Also the vein system, the veins means these veins, right? The veins which you see inside. And how are these veins arranged? The veins can be reticulate arrangement or parallel arrangement. Reticulate arrangement means net-like design, if you can see them with the magnifying glass. And the reticulate venation is seen in all dicotyledonous plants. And another veins which are seen in the paddy fields and the other fields, monocotyledon, parallel arrangement. So this is a very important question, that what are the features found in the leaf which helps in maximum photosynthesis? Your answer should be surface area. Your answer should be the arrangement of the leaf, presence of a thin cuticle and epidermis, then lots of stomata, then the extensive vein system. This is the first adaptation. Next internal feature which governs it is the presence of chlorophyll. Told you that. Okay, and what are the external features which affects photosynthesis? Temperature, light intensity, amount of CO2 available, amount of water available. Okay, but CO2 plays a limiting role in the rate of photosynthesis. Intensity of light, I told you, blue light, red light, maximum. And no photosynthesis in green light. Yes? Okay. So external as well as internal factors governing the rate of photosynthesis, if they ask you that. After this, now we come to, so what you will write, if they make a column and write, for the internal factors, I repeat again, chlorophyll, which is the content of the leaf. Second, the structure of the leaf, told you. Third, the protoplasm. Dehydration of protoplasm. If the protoplasm gets dehydrated, very important, it reduces the rate of photosynthesis. Then, Okay, and the accumulation of end products. The progress of photosynthesis in the green cells is maintained as long as the concentration of the products formed. Which products formed? The sugars, and the starch formed is minimum. So these are the important points to be written for the internal factors. And for the external factors, I told you light, CO2, temperature, and the amount of water available. So this was your previous knowledge, background of this. Now we come to how does this photosynthesis, what are the events which takes place, okay? So I will tell you two important events here. As I told you, the light reaction and the light reaction and the dark reaction. Now these two reactions, they take place simultaneously in the plant. It doesn't mean that this is light dependent, it will take place when no light is there. No, nothing like that. They take place simultaneously in the plant. Only the site is different. This is grana, this is taking place in the grana, and this is taking place in the stroma. Grana and stroma. Okay. Now take a notebook and please write what I am just telling you, the important things here. First you will write here for the light reaction. This is a light dependent reaction. First point, light dependent phase. Second, here it is initiated by quick chemical reactions which is initiated by the light, and that is why it is called as photochemical reaction. It takes place in the visible wavelength 400 to 700 nanometer, that part of wavelength where the thylakoids of the chloroplast absorb the light, 400 to 700 nm. This is the wavelength, visible spectrum of light where photosynthesis takes place. After that, what are the three steps here involved? First is activation of chlorophyll molecule. The chlorophyll molecule gets activated. From the ground state, it comes to the excited stage by absorbing one photonic energy of the sun. It gets activated. That is, the chlorophyll molecule gets activated by absorbing the photons of the light energy of the sun. Activation of chlorophyll molecule is the first step in the light reaction. After this, what happens? Second step, splitting of water molecules. Splitting of water molecule. That is, the splitting of water molecules, 2H2O splits into 4H plus 4 electron plus O2. Okay. Now, splitting of water molecule. This splitting of water molecule is known as photolysis. Photolysis is the key word. Photo means light and lysis means breaking. So breaking of water molecule. Now what happens with the end products of this water molecule? What happens here? Here what happens, this 4H is accepted by compound NADP. Name is given in your book, nicotinamide adenine diphosphate. And it forms NADPH. Nicotinamide adenine diphosphate. Okay, it gets reduced after accepting the hydrogen. And what about this 4 electron? This 4 electron, this is further used for making of ATP by the term given as photophosphorylation. Photophosphorylation. That is ADP, adenosine diphosphate, plus one inorganic phosphate, plus this electron gets transformed to ATP. This is termed as photophosphorylation. Break it. Photo, addition of phosphate, phosphorylation. That is, it is the process in which the ADP, adenosine diphosphate, is converted to ATP by the addition of phosphate and also by the taking in of the electrons which is produced here. And this ATP is further used in the dark reaction. So this is a very, very important thing which I have told you here, and that is activation of the chlorophyll molecule. photolysis of water, and photophosphorylation. So in summary, if you summarize it, it's given on your book. See the light independent reaction and the light dependent reaction. Thylakoids on page number 73. Your ICSE book, hydrogen, electron, oxygen. Oxygen goes to the environment, to the outside atmosphere. It is taking place in the thylakoids. So hydrogen is taken by the NADP. NADP is reduced to NADPH. It's given here, and this NADPH will be further used for giving hydrogen for making of sucrose, glucose in the dark reaction. It is given here. Photophosphorylation term is also shown here. ADP, how ADP plus P makes ATP. So in summary, light reaction involves conversion of light energy to chemical energy in the form of ATP and NADPH molecules. Got it? Light reaction. First part of photosynthesis in the thylakoid. Now what about dark reaction? What is this dark reaction? So you have to write, please write. Dark reaction is a biosynthetic pathway. It's also known as the Calvin cycle. Biosynthetic pathway, also called as the Calvin cycle. Now, the phase in which synthesis of organic compounds takes place is known as a biosynthetic cycle. The term dark reaction doesn't mean that the reaction occurs only at the night. It is light independent, which means in the presence or absence of light. Got it? And what happens here? 3 events takes place here. Those 3 events are, first is carboxylation. Carboxylation. What is this carboxylation? It is the most crucial step of this dark reaction or Calvin cycle, in which the utilization of carbon dioxide takes place for the carboxylation of RuBP. RuBP is ribulose biphosphate. It's the organic molecule. RuBP in presence of an enzyme Rubisco, and RuBP gets carboxylated. CO2 plus RuBP plus water forms a molecule known as phosphoglyceric acid. 3 molecule, 3 PGA is formed by phosphorylation of RuBP. If you want to write the reaction, CO2 plus RuBP plus water will give rise to 3PGA. 3PGA. This is the carboxylation. It takes place in the stroma. First reaction. Then after this what happens? Reduction. Carboxylation takes place here. Second step is reduction. Reduction. And third step is regeneration. RuBP is returned back. It is formed back again, so that again the cycle continues. Reduction of what? Reduction of this PGA to what? To form glucose. That is known as reduction. After carboxylation reaction, the reduction of PGA takes place in a series of steps where glucose is formed. So just remember, dark reaction stroma, 3 steps. Carboxylation, where the acceptor of the first accepting molecule is RuBP. RuBP is accepting it to form 3PGA. Second is reduction. Reduction of this PGA, phosphoglyceric acid, by a series of events which is known as Calvin cycle, to glucose. And third is another complicated reaction where RuBP is formed back again. Okay? So a comparison of light reaction and dark reaction should be very clear in your mind. So till now, what is photosynthesis? Structure, chloroplast. What is mesophyll tissue? What is mesophyll tissue made up of? How does stomata open? What is the mechanism of stomata opening? 2 theories, potassium theory, sugar theory. After that, the factors which affect photosynthesis, leaf and all I told you, external and internal. Then after that, told you the 2 phases of photosynthesis, the light reaction and the dark reaction. I think I have given you a brief picture of the entire lesson, which in last minute should be there in your mind. Now you come to your book and see the last part of the lesson. The last part of the lesson is some experiments which are very important, which you will draw and write and understand the importance of photosynthesis by the writing of the aim. Your teacher should show you this. First one is, the first experiment is where they have shown you removing of the chlorophyll, de-starching of the leaf, iodine test. It is asked in exam. And what is the role of iodine? What is the role of hot water bath? First experiment is that. Second, you come to page number 76, where they have shown you to show that sunlight is necessary for photosynthesis. They are blocking it with a black paper, and then the rest area, it gives the starch test. Why? Because that is not exposed. Third one, to show that carbon dioxide is necessary for photosynthesis, is this experiment which you should study, where they have taken a destarched leaf, then the entire procedure is very clearly given here, and you will see that the carbon dioxide is necessary for photosynthesis. After read and draw and write the experiment. Fourth one is to show that oxygen is produced during photosynthesis. They have taken a single very nice experiment. This is, they have taken a hydrilla plant in they have inverted a test tube over it, and then they have kept it for a few hours in the sun. Bubbles of gas will collect over the test tube. So oxygen is evolved as a product here. Right? So these are the three experiments in your mind. And lastly, what is the importance of photosynthesis, LMR? First, it provides you food, because plants are autotrophs. Plants are the nectars of life. Without them, they are the starting point of the food chain. And what is the food chain? Where the one organism is being eaten by the another organism in a sequential manner. Food chain and food web all depend upon these plants, which are known as the producers, and they form the first trophic level in the entire ecosystem of this entire biosphere. Second is, it provides oxygen, right? And the third importance, combining both photosynthesis, food chain and everything, comes the carbon cycle, and the figure is very clearly given on page number 78. So I think LMR of this lesson is clear. If you will listen, because the subject is learned by reading, writing and speaking. I spoke to you about the lesson, photosynthesis. I showed you, at the end, I conclude my video now, I showed you this leaf. Again I show you this leaf, which is of three types, yet the common feature between them is photosynthesis. And what it is, where it takes place, the stages, the components, the importance, the factors, and in the end, the carbon cycle. Now you have to read the lesson on your own. By keep this video on, it will give you some data in your mind. I am not becoming much complicated. Hope you have liked my presentation. Keep seeing, stay geeky, and keep subscribing. And thank you. Thank you, students.